All right, so I wanted to make a little video for those of you that are trying to figure out what to do with this DNA coloring assignment. So really all you have is this page. Now, granted, if you're in school, this will be so much easier. I can explain these things and show you and give you some examples. But right now, it creates some difficulty being at home. So I wanted to make a little video. You're basically going to get this. And so what are all these little things? Well, this is showing you a bunch of different nucleotides, okay? And so we talked in class about the parts of a nucleotide. Let me just zoom in here on this guy. So you see the phosphate group, that's this P. You're going to color that a particular color. This is a sugar ring. You see that little pentagon here. This is the sugar. And then you have the nitrogen base. This happens to be cytosine, okay? Thymine, and as you go down, adenine and guanine. And we said that these bases are what change on a nucleotide. The phosphates and sugars stay the same. You'll notice they stay the same throughout, okay? These bases are what determine the code. And so what you're going to do is you're going to create a complementary strand of DNA from your original strand. So basically, you're going to mimic what your body does when it copies DNA. So the first thing you'll have to do is you are going to need to take this strand of DNA, GAGTC, and you're going to want to snip this and then paste it. So you're going to go down here, find the snipping tool, right? Type in snip, find the snipping tool. I'm going to do a new snip, and I'm just going to snip this guy right here. That's all I want is just this little guy right here. Okay, right there, perfect. Then I'm going to click edit and copy, and then I am going to paste that in a Word document. Okay, why are we pasting it in the Word doc? This is where all your drawing tools are. Okay, then the next thing you're going to do, and you'll probably want to shift this down a little bit. We'll see why in a minute. Okay, so we have our original strand, then you need to come up with a complementary strand. So we're asking you, if this is the guanine nucleotide, what would bond with it? Well, we learned in class G always pairs with C. So you're going to grab a cytosine. We'll go back to our document here. You're going to grab this cytosine nucleotide and you're going to snip that. You're going to click new and you're going to just snip just enough to get cytosine in the picture. Okay? You click edit, copy, and then we'll go back to our Word doc and we'll try this. Paste it in there. Let me see if we can drag this guy. Let me see if that helped at all. It doesn't look like it. There. Okay. So that seemed to work. All I did there is I went through and I clicked text wrapping and I selected this option through. Okay. So select this. That'll put it so that you can move this around easier. Don't get it too close. Otherwise, it'll want to cut it off. You'll notice, though, how does this guanine bond with this cytosine? Well, it can't. And that's part of this activity. You'll notice that the only way to bond this G and the C is to flip this upside down like that. And then you can get it in, I got too close, get it in close there. And that's where they'll bond, okay? Half of the point of this activity is not only to see the structure of DNA, but to understand that one of your strands is quote unquote upside down or backwards. And that means something when we go to read this stuff and copy this, okay? So you're going to want to do this for this one. Then you're going to need to go through and grab the next one. What was my code here? I had an adenine, so I need a thymine over here. And you'll notice that this C fits perfectly with the G. The A will fit perfectly with this T here. I'm going to snip this T. New. And again, all I want just barely want to fit this in there. Okay? I'm going to click edit and copy again. I'm going to go back And I'm going to see if we can add this guy in here. Paste it in. That sent us all haywire. I'm going to go over here and I'm going to click through again. And hopefully that helps there. It did help. Okay. But again, I have to flip this guy upside down, and I'm hoping that we can get it close enough. Oops, too close. Just like that. Now, the only problem is you'll notice this. We're not showing the bonds. You're going to want to show those. Okay. I don't have a preference what you color your phosphates, but be consistent and make me a little key, enter the text box, color your sugars the same, and then whatever you want to color guanine, maybe green, color all of your G's green. Okay, whatever you want to color adenine, color that whatever, but just don't duplicate any colors if that makes sense. You'll notice that your sugar phosphate backbone isn't attached. You have to attach the phosphate from this one to the sugar from the next one. So all I want you to do here is you're going to go insert, and you're going to insert a shape, or you can just click the drawing toolbar, it doesn't really matter, and you're going to attach this phosphate right there. Okay, I'm going to select black, and I'm just going to make it a little bit thicker, go to the weight, and maybe make it something like that there. Okay, just to show that those are connected. All right, you're going to do that all the way through, and then color code it, and then you're going to email this to me. Okay, that way I can see that, or message it to me in Canvas. That way I can see that you've done it. Okay, so that's what you're doing. Use your drawing. I'm going to go, let's maybe, and I'll use the highlighter, of course. Maybe I'm going to use purple for phosphate. That's purple-ish, right? Use my purple for phosphate. Color all my phosphates purple. I probably should have picked a smaller thickness here. Maybe I want to do all my sugars. Maybe I'll do all my sugars blue, maybe. Maybe I'm going to do all my guanines green. Okay. Color this guy in. And the last thing you're going to do, let's say I'm going to pick cytosine. I'll do that one in orange. The last thing you want to do is you need to show the bonds between the G and the C and the A and the T. And if you remember, I'm going to just do a line here. It's easier. If you remember, and I'll do red. G and C bond 3 times. 1, 2, and 3. A and T only bond twice. 1, 2. And then just make sure you know what these are. Make sure that you know that those are those hydrogen. I don't have a stylus here or a touch screen. Hydrogen bonds. Okay, make sure you know those are hydrogen bonds. And then make sure you know that this is your phosphate, this is your sugar. Okay, this model is just supposed to help you. 1, understand the structure of a nucleotide. 2, understand how they bond. And 3, understand this last idea that one strand is always upside down or backwards, and that affects how we copy it. Okay, so you're going to complete this, snip those things out like I did, color this guy, make sure you select that through button so that it doesn't try and move it to the next screen or move it off somewhere else. You guys should be good to go. Okay, if you have questions, let me know.